Hello, welcome back to our look at part 6 of biochemistry, specifically looking at the biomolecules. In the last video, we spent some time looking at carbohydrates, and I just want to kind of outline that for you just to kind of draw your attention to a few things. First thing was for our biomolecules, we had carbohydrate. And we said that a monomer of a carbohydrate was a monosaccharide. And when these monosaccharides undergo dehydration synthesis, we said that they form a glycosidic bond, and that glycosidic bond was created between two hydroxyl groups. Right, that would then go ahead and remove water. In this video, we need to look at and understand the relationships now that we talked about within carbohydrates. We need to start applying them to our second biomolecule group, which is nucleic acids. And so at the end of this session, we're going to kind of come back and fill in the monomers for nucleic acids, what kind of bond that they create, and what they are joined, what that bond is really joined, created with. And nucleic acids are a little bit of a special beast, but we'll get into that. And so let's go ahead and let's start our look at nucleic acids. All right. First of all, nucleic acids are critical in creating energy. Right, cellular energy, both ATP and GTP. Right, although ATP typically gets the majority of the glory and the fame, ATP is not the only type of cellular energy out there. As I said, we also have GTP. And so ATP and GTP are types of nucleic acids, but so are DNA and RNA. Right, so our DNA and or RNA and DNA, and so these are the types of things that we're going to be talking about during this segment of our recording and videos. A monomer of a nucleic acid is what we define as being a nucleotide, and so a nucleotide is a monomer of a nucleic acid, and all monomers have a very similar structure. All monomers have a very similar structure. They all have a phosphate group. Right? And that phosphate group never changes. It is consistent. Right? That phosphate group is consistent. All right? So it's always the functional group that we learned as being a phosphate group. The next thing that all nucleotides have is all nucleotides have a 5-carbon sugar. It's got a pentose. Right? And we know that we learned that there are 2 types of pentose sugars. There is ribose and there is deoxyribose. Right? They are the 2 types of sugar. Now, we also know that from our look at carbohydrates that how we differentiate ribose from deoxyribose is by looking at that 2' carbon. Right? That is what defines whether or not we are dealing with ribose or deoxyribose, and which will ultimately define whether or not we're dealing with deoxyribonucleic acids, or DNA, or ribonucleic acids, or RNA. And so you got some variability there. Right? You can either have ribose or deoxyribose. And then you have a nitrogen base. Right, then you have a nitrogen base. Now, there are 5 possible nitrogen bases that you can have in this spot. So you have a lot of variability here. All right, now, some of that depends on the sugar. All right, some of that depends on the sugar that is bound here as well. And we're going to talk more about that in a little bit. But right now, I'm going to bring you back to your high school biology days and say that those nitrogen bases can either be A's, T's, G's, C's, or the letter U. All right. It can either be an A, a T, a G, a C, or a U, letter U. All right. So they are your choices for nitrogen bases. Now the question is, how do we go from this nucleotide, which again is the monomer, how do we go from there to more complex structures? And the answer to that is twofold, because really nucleic acids are engaging in two different types of bonding. We're going to talk about the first type of bonding now. All right, so here are two nucleotides. All right, here's the phosphate group. Here's the sugar. We look at that 2' carbon and we see that it's missing an oxygen, so therefore we know that we are dealing with deoxyribose. Right? If you wasn't sure about that, it says it up there. Right? And here's the nitrogen base right here. This is adenine. Right? This is adenine, and so this is an adenine nucleotide. And adenine always is bonded with thymine. And so here's the nitrogen base here. We can see it's labeled thymine. Here's the sugar. Okay, we can see that it's missing the oxygen, so therefore we know it's deoxyribose. Right? And here's the phosphate group right here. And you will notice that the adenine and the thymine are being held together by these two bonds right there. Those two bonds, as indicated by the H and the H, are hydrogen bonds. And when we are dealing with DNA, adenine and thymine will always create a double hydrogen bond, while guanine and cytosine will always create a triple hydrogen bond. All right. So that is one way in which we can bond nucleotides. Right? We can bind these nucleotides based off of their nitrogen base. Right? And we can create these lateral bonds. Right? That's really what the hydrogen bond is. It's a lateral connection of our nucleotides. Right? It's a lateral bond of our nucleotides. But we can also create bonds. We can also create bonds vertically. And so in other words, there's another nucleotide sitting here. These two get connected. There's another nucleotide sitting here. Those two get connected. And we're going to look at that specifically on not the next slide, but the slide after this next slide. Because I want to show you how this is all fitting together. Because we've got horizontal bonding and we've got vertical bonding going on. And so when we do the horizontal bonding and this vertical bonding, we create what you all characteristically know as being DNA. And you can see wherever there is a C and a G, there are 3 bonds. Right? But whenever there is an A and a T, there are 2 of those hydrogen bonds. Right? And you can go through this whole thing like that. Right? Every time you see a G and a C, there's going to be 3 lines. And every time you see an A and a T, there's going to be 2 lines. T and A, 2 lines. G and C, 3 lines. So they are, again, they are double and triple bonds. All right? So lateral bonding is going to result in double and triple hydrogen bonds. All right. Double and triple hydrogen bonds. That's lateral bonding. But what about vertical bonding? What about vertical bonding? Well, let's take a look at this. All right. This slide here shows how exactly we do vertical bonding. So let's take a look at this. If we look at this image, here is the sugar. Right? Here's the sugar. Right? We're going to look at the 5' carbon, because this is a pentose, so there's only 5 carbons, and we see that the phosphate group is attached. Right? We see that this phosphate group right here is attached. But this phosphate group is also attached to the 3' carbon of the nucleotide above it. Right? Of the nucleotide above it. So here's one nucleotide. Here's the nitrogen base. Here's the sugar. Here's the phosphate group. Here's the next nucleotide. Here's the nitrogen base. Here's the sugar. Here's the phosphate group. Here's another nucleotide. Here's the nitrogen base. Here's the sugar. Here's the phosphate group. What you're not seeing here is that those nitrogen bases are actually forming double and triple bonds. So this is thymine, so this would actually be forming a double bond with an adenine nucleotide over here. This is cytosine. It's creating a triple bond with guanine over here. All right, this is guanine. This is actually producing a triple bond with cytosine over here. So that your horizontal articulation is still happening. But this is vertical. This is now attaching them up and down. And again, this is happening between the 5' carbon of one sugar with the 3' carbon of the sugar above it. And what we are using to create the bond, what is actually removing the water, because water is being removed from in here. Right? Water's being removed. Right? Because remember, this is an OH. Right? Remember, there's an OH over here, attached to this carbon over here. Right? So we are still, and remember that these guys both had charges. Right? They both had charges on them. And so that hydrogen ion gets transferred, and you are creating and releasing that water still. The bond that is formed between the phosphate group and the 5' carbon of one sugar and the 3' carbon of the above sugar is what we identify as a phosphodiester bond, or sometimes you will see it referred to as a phosphodiester linkage. Right. And think about the name, phospho, because it's involving a phosphate group as the intermediary. Di, which means two. Ester, which is the bond that is created between the oxygen and the carbon. But why is it diester? Because you're creating 1, 2 ester bonds. So you're using a phosphate group to create 1, 2 ester bonds that then join vertically our nucleotides. Now, I will mention that you don't need to know it in that level of detail for the exam. For the exam, I do expect you to know that nucleotides form phosphodiester linkages, right, or phosphodiester bonds. Right? I do expect you to know that the phosphate group is what coordinates that. Right? I do expect you to know that that bonding is a vertical articulation, and that when we talk about lateral bonding within nucleic acids and nucleotides, it is a hydrogen bond that is formed, and when it's an adenine and a thymine, or an A and a T, it's a double bond, and when we're dealing with cytosine and guanine, it's a triple bond. All right? So that I do expect you to be able to know. All right? And we're going to summarize this at the end as well, so don't get overly freaked out. I've already kind of mentioned this. I'm going to mention it again just because redundancy is always a good thing. And that is there are two main types of nucleotides that are being created using our nucleic acids, or that are used to create the nucleic acid. So our two types of nucleotides are ribonucleotides, and ribonucleotides use ribose. And then we also have deoxyribonucleotides, which uses deoxyribose as the sugar. Right? And again, I point to here the 2' carbon because this is where the difference is. Other than that, these guys are identical. Right? These are identical. There is no difference here. The only difference is the OH and the H. Deoxyribose on the 2' carbon is lacking in oxygen, as its name implies. Deoxyribose. All right. Okay. So, nitrogen bases. I want to spend a few minutes talking about nitrogen bases, and this is going to be something that we're going to be revisiting in Chapter 3, because we're going to talk about DNA replication. And so understanding it now will help you more in Chapter 3. But nitrogen bases really determine the type of nucleotide that is created. So once we know whether it is a ribonucleotide or a deoxyribonucleotide, what specifically are we dealing with? Are we dealing with an adenine nucleotide or a thymine nucleotide? And what determines that are the nitrogen bases. All right. And I've already kind of hinted at this. You see the A, the G, the C, the T, and the U. All right. So A stands for adenine. G stands for guanine. C stands for cytosine. T stands for thymine. And all four of those are always found in DNA. And then in RNA, we replace thymine with uracil. And so in RNA, you have adenine, guanine, cytosine, and uracil. Right? In RNA, there is no thymine. And we're going to look at this a little bit more. So if that wasn't quite making sense, it will make sense once we start going into this in a little bit more detail. And what you need to understand is that adenine always bonds with thymine. Guanine always bonds with cytosine. Right? And this is a lateral bonding. We're not defining vertical bonding. We're not defining the phosphodiester linkages. We're talking about the hydrogen bonding. Right? Moving left to right. All right, moving left to right. And to help coordinate this, to help keep this straight in our minds, we actually can classify a little bit further the A's, the G's, the T's, and the C's. So we have two types of nitrogen bases. We have our double nitrogen ring structures, like what you see down here. All right, so here's our double ring nitrogen structures. We call them double ring because there's 1, 2. 1, 2. So guanine is a double rung structure. Adenine is a double rung structure. And we refer to them as purines. Right, so both guanine and adenine are purines. Right, double nitrogen rung structures. Cytosine, uracil, and thymine are what we classify as pyrimidines, single rung structures. Single rung structures. All right, pyrimidines. So pyrimidines, cytosine, uracil, thymine, because they are single nitrogen ring structures. Guanine and adenine are purines because they are double rung structures. And if you pay attention to the color coding, guanine always binds to cytosine. Adenine binds or bonds to thymine. Guanine bonds to cytosine, but adenine pairs. It does not bond to uracil. It simply pairs with uracil. Again, that'll make more sense in chapter 3 why that happens. All right. So I want to spend a few minutes talking about RNA. I'm not going to spend a lot of time on this. I really just want to kind of mention the fact that RNA is less stable. As a type of nucleic acid, RNA is less stable. It is more readily to be dissolved by water than DNA. DNA is very stable. And really what allows for DNA to persist, what allows for DNA to not dissolve in water, because it is surrounded by an aqueous solution, is the fact that it is missing the oxygen. This lack of oxygen right here makes it less reactive with water, and so it stabilizes the DNA. So you don't have to worry about your DNA dissolving. It's going to remain intact, which is a good thing. Right? RNA, on the other hand, is made with ribose. And the reason why it is less stable is because of the presence of that OH group. This OH group makes this more reactive in water, and so it's more likely to be dissolved in water. And if we think about the function of RNA, RNA is used to create proteins. Proteins are only made as needed. So we don't need RNA to be stable. We need RNA to persist long enough to make the protein and then be broken down. And so it makes sense that RNA would be more reactive in water, right? More likely to bond with water and then have that water basically disassemble the RNA. And then we can recycle the parts. But DNA has to persist. If your DNA begins to break down and dissolve, what ends up happening? You break down and dissolve. And that's not good. We don't want that for anybody. Okay, last topic that I want to discuss with you all about nucleic acids, and that is a special case, a special scenario of a nucleotide. Right? This is a special case of a nucleotide, and that is adenosine triphosphate. This is our big major energy producer within the body. And so its role is to drive metabolic reactions. This is what allows us, this is what allows our cells to function. This is what allows your muscles to contract. This is what allows action potentials through the nervous system to be generated. This is what allows us to go ahead and survive on a daily basis. This is cellular energy. And so you can kind of see here the overall structure and what we use for this. The key thing to remember here is we're dealing with an adenine molecule that is bound to a 5-carbon sugar. All right, and that 5-carbon sugar is ribose. Adenine plus the ribose is what we identify as being the adenosine. All right, so when you see that adenosine term, adenosine means that the adenine is bound to ribose. All right? That is what adenosine is. Right? And then adenosine is attached to phosphate. 3 phosphate groups. 3 phosphate groups. And what is connecting those 3 phosphate groups are hydrogen bonds. High energy bonds that break very easily because of the amount of energy that they're containing. And so what ends up happening is that 3rd phosphate group pops off. And when it pops off, it releases this energy. Now, that stabilizes the structure. By the way, you no longer have adenosine triphosphate. After the 3rd phosphate, after it pops off, how many phosphates do you have left? 2. And so now you have adenosine diphosphate. And there's still more energy in that bond. It can pop off. And when it does, it releases some more energy. Right, so let's look at that organization. Right, let's look at that organization. So you see right here in this diagram that we are dealing with here's the nitrogen base. That's the adenine. Here's the ribose. We know it's ribose because it's got the OH. Right? And so these guys connected is the adenosine. And then we have 1, 2, 3 phosphate groups. Triphosphate. Right? Triphosphate. And so when we engage in hydrolysis, when we add a water molecule back in between the 3rd phosphate and the 2nd phosphate, this guy pops off. Right. And so what we then are left with is ADP, adenosine diphosphate. Right. Adenosine diphosphate plus an inorganic phosphate group because this guy is free floating now. All right. We can add another water molecule back in here and break the bond between the 2nd and the 1st. He now pops off. And now what are you left with? Adenosine with 1 phosphate group. It's a adenine nucleotide, or what we also refer to as AMP, adenosine monophosphate. Right. But what's amazing is that we can actually go through and we can reverse the process. We can actually add phosphate groups back on by doing dehydration synthesis. And so we can actually take this phosphate group and add it back. We can take this phosphate group and add it back. And the process of adding a phosphate group onto a molecule, in this case adenosine, is called phosphorylation. Phosphorylation is the process of adding a phosphate group back onto a particular molecule. In this case, we're adding it onto adenosine. So we can add a phosphate group onto AMP and now have ADP. We can add a phosphate group onto ADP and now have ATP. And this is exactly what happens in your muscular system. This is why a lot of those post-workouts and pre-workouts, you're going to see certain terms in there, right? Like creatine. What the hell's creatine? What? Creatine phosphate? What the frig is this? We're going to talk about that. But what I'm going to tell you is that is what aids in the direction of phosphorylation in your muscle cells. that allows you to produce ATP to drive protein synthesis, which drives recovery, builds muscle mass, allows oxygen to get in there quicker to do more recovery. These are the real world applications we need to look at. Right? So, good news, bad news. We've got this process called phosphorylation. Right? That's the good news. The bad news is there's different types of phosphorylation. You didn't think I was going to let you go that easy, did you? All right. So you've got we've got three basic types. Right? We have photophosphorylation. Right? So this is adding phosphate groups back onto molecules using sunlight. We don't do that. Plants do that. We don't do that. Right? No, it exists. We don't do it. So we're not going to ever talk about that. All right? We have substrate level phosphorylation. This we do. Substrate level phosphorylation uses enzymes to drive the production of things like ATP. Right? We're going to talk about that in Chapter 3. I'm going to give you some very specific examples of substrate level phosphorylation, right, where we're adding phosphate groups onto molecules, right, with the use of an enzyme. And then we have oxidative phosphorylation. Shoot. Completely lost it. Sorry about that, guys. Photophosphorylation. I'm just going to rewrite this real quick for you. Which again, we don't use substrate level phosphorylation, which again uses enzymes. And then we have this third type, oxidative phosphorylation, which uses hydrogen ion concentrations to drive the production of ATP by adding phosphate groups back onto molecules of ADP. But we're using hydrogen ion concentrations to do that. And we're going to talk about that. That is, again, that happens during cell respiration. This is a Chapter 3 type of thing. All right, so just know that there are different types of phosphorylation that you're going to be responsible to apply and know and understand. This here just kind of goes through and just shows, again, that structure of ATP, right, and why it is a special case of a nucleic acid. Right, here's the nitrogen base. Here's ribose. 1, 2, 3. ATP. Right, but what happens if we take away an ATP molecule? We release energy, but we're also down to ADP. Right? What happens if we take away this phosphate group? Well, we released more energy, but now we're down to just a regular old adenine nucleotide, right, with the nitrogen base, the sugar, and the phosphate group. All right? We can add it back through phosphorylation. We can add it back through phosphorylation. And then we can release more energy. And so we can actually continuously build and rebuild and use and rebuild these molecules. I will say, ATP is not an effective storage of energy because it is highly unstable because of the amount of energy that it reserves. The body can only hold about 30 seconds of ATP in reserve. That's it. All right, that's it. And so we constantly have to be finding ways of making new ATP. And we're gonna flesh out some of those ideas as we go throughout the semester. All right, and this diagram here just again kind of shows us that relationship. All right, what happens when we only have adenine and ribose? We have adenosine. All right, if we add one phosphate group, it's AMP. If we add two phosphate groups, it's ADP. And if we add that third phosphate group, it is ATP. Okay, so now let's go back and let's fill in this table. All right, once again. And we will review this each video beginning and end just to help you guys keep things straight. So now we have, we started with carbohydrates. And we said that its monomer was a monosaccharide. We said that the bond that was formed was a glycosidic bond. And we said that that glycosidic bond was created between two hydroxyl groups. And then we just got done now talking about nucleic acids, for which we said that their monomer was a nucleotide, and the type of bond that they can create is both a hydrogen bond, and again that's lateral articulation, but more importantly the vertical articulation is a phosphodiester linkage, right? And that is created between a phosphate group and the 5' carbon and 3' carbon of adjacent sugars. All right, and so that's kind of where we are right now. I think this is a lot for you to go back and review, and so I'm going to give you some time to do that. Remember, continuously jot down your questions, reach back out if you have questions, and I'll catch you on the flip side.